Hi, okay, so this is the Photosynthesis lab by Sunny and Jake. Our question that we tested was, how will the ET50 time change in the leaves of spinach if we change the concentration of bicarbonate? Our hypothesis was that the higher the concentration of bicarbonate, the faster the ET50 time will be.
Are you videoing this?
Yes.
You're videoing me?
Live action of the hole punching of the 10 discs for the first trial, which is the control run. Jake is currently using the hole punch to punch out the small discs out of the baby spinach.
Tell me when you're ready.
Good.
There's like
Vacuum kind of.
Do I just hold this here?
What is this called?
Make it a vacuum?
Making vacuum. So the discs sink to the bottom, and
Now they're floating.
All 10 discs have sunk. Have just put discs under lid. We are at the 5-minute mark. Still no floaters.
6 minutes. Go.
Grace is being very sarcastic and thinking she is helping us. This is for the vlog. Update. We are redoing the first trial, the control group, because this cup was contaminated by baking soda, which led to the oxygen being trapped at the bottom. And originally we thought that we had done something wrong because none of them rose to the top. But once we moved the cup, all of them rose to the top, so that's how we can conclude that the oxygen was trapped at the bottom. Trial number 2.
3.
Well, 3 kind of, counting the control.
Actually, it's like more like 4.
This is 2 times the bicarbonate.
Okay, bye.
Yes. Have just put discs under lamp. Ruler was not needed. Phone is dying, but we're on the second trial, which is double the bicarbonate, and they are floating much faster. It's only 5 minutes in, and we had 8 that have risen. So, data shows that the water was the slowest, the regular bicarbonate was in the middle, and the double the bicarbonate is the fastest and most photosynthesis producing.
Okay guys, here we can see our ET50 times for each trial. As you can see, the times decrease as we introduce more carbonate into the system. This was expected with our hypothesis because we figured that when you introduce more carbonate into the system, the plants will take in more CO2 and photosynthesize quicker. And 2 constants within our experiment are the temperature and the light that we use. Okay, update number 1. I went ahead and made a histogram showing to better show the ET50 times for each trial, starting off with control, trial 1 and trial 2.
Our sources of improvements would be to make sure that we cut the leaves, the discs, specifically not close to any veins, and we can make all 10 discs as similar and as accurate as possible. An explanation of divergent results. Actually, our very first trial that we did was completely off and didn't seem right. And we figured out that was because the cup we were using had leftover residue from a substance that was in there before. The discs this is our explanation of findings. The discs photosynthesized because they absorbed the CO2 from the carbonate, and the more carbonate we added made the concentration higher. They were able to absorb more CO2 and go through photosynthesis quicker because of the added CO2, and which is why they gave off more oxygen, which led to the lower ET50 time and the faster rising of all 10 discs.